opposite direction all the way through, right? So all 8 strands. Make sense? And then the alpha helices themselves are doing what? Just 1, 2, 3 alpha helices in this thing? Right? This one's kind of a, you can call this one if you want, but it's really because of this disulfide bond, they're kind of broken into here, right? This disulfide bonds and proteins and other things don't work well on alpha helix. But you see how they're kind of holding the sheet. And so they can interact with each other. Here's another example of nothing but beta strands. In this case, because of that propeller twist, they're twisting on each other to make a cylinder. That's called a beta barrel. Make sense? So it's not as simple as just strands next to each other or a helix. If you start looking out larger, you get bigger structures like this. But these are all secondary structures. Alpha helices, beta sheets are all secondary structures. All right, so here's the third thing that a secondary structure could be. If it's not an alpha helix, it's not a beta strand or a beta sheet, it's called a loop. Kind of a generic either of the other two. This is called a loop. It's just an unorganized piece of the protein. That's what they're called. Just loops. Some loops are called terms. Right? This is like saying, I'll give you an analogy for this, right? Loops represent fish. Some fish are sharks. Right? Every shark is a fish, but not all fish are sharks. Right? It's a subcategory. That's what this means. All terms are loops, but not all loops are specifically terms. Do I see the difference in the hierarchy here? So anything that's not an alpha helix or a beta sheet, these guys here, right? Every one of these things here in this top part, or these all down here, would be considered loops. If I scroll back, these would all be considered loops. Right? All these things would be considered loops. Well, not that part, but all this. All the unorganized stuff, right? Some of those loops are very, very short, like this one. Right? All it does is connect these two ends. Right? So it'll be considered a term. So it's really short, like maybe this guy right here. Right? It's just a term. This is not short. This is quite long before it goes back to something else. That's just a regular old loop. Okay, so really short loops that connect two other elements, right? Usually beta strands or alpha helices are going to be called terms. Just a small loop. That's all it is. Some of those terms are called beta terms. You see the hierarchy, we get more specific. Right? So a beta term would have what qualification? Well, it must first be a term. So it's a really short loop that connects two things. But what two things does it always connect? It has to be strands of an anti-parallel beta sheet. Why can't they be parallel? Yeah, they have to go in opposite directions because if one ends and it turns to the next one, like that little one right there, if they were going in the parallel direction, it'd have to end, come out here somewhere, and end up going this way. It can't be short. Right? So there's no way you can have it between parallel strands. So beta terms connect anti-parallel strands. Right? All of which are considered terms. Some of these connect alpha helices to strands, or strands to, or two alpha helices, or some other secondary elements. And then all of those terms are considered loops, and many loops are just unorganized, things like that. Okay with that? So there's three main secondary groups, right? Alpha helix, beta sheet, or loop. There are some subcategories of loops. That's all this is saying. What holds all the secondary structure together? Hydrogen bond. There's going to be far fewer of those in these terms, right? You see, this only has one hydrogen bond. This is one of those terms that turns around. There's only one hydrogen bond. Okay, let's practice real quick at the bottom here. So let's compare which one of these numbers goes with these letters. It's easiest just to read the description and choose a number. Okay, so A says leucine and valine could interact how? Which one of those interactions could they do? They're only hydrophobics, right? So there are groups who really don't do much else, right? They're just hydrophobic R groups. They can't hydrogen bond, they can't make a disulfide, and they're certainly not charged to do the ions. So yeah, just hydrophobic. How about two cysteines? Disulfide, right? Aspartic acid and lysine. Yeah, this is an acid, so it will deprotonate and become what charge? Negative, and lysine is one of your bases, so it's going to gain a proton at a pH that's more acidic than its pKa and become positively charged. So this is an ionic interaction. And serine and threonine, both of which are alcohols, so they're gonna interact in which way? Hydrogen. Now, that's not to say that these could not hydrogen bond, right? But most likely would be ionic interactions from this. But these would certainly be hydrogen bond. Threonine could have some hydrophobic interactions. There is another group there, but not with serum. Right? So these two can only do that. So it's easy to pick out which one's which. Okay, so here's all the different shapes we have for tertiary structure, right? So it's an easy way to think about it. If you zoom out and look at the whole protein, it's either one of three shapes, right? In broad categories. It's either going to be spherically shaped, roundish, doesn't have to be a perfect basketball, but it's have a round shape, right? This is called globular. Makes sense, kind of like a globe, globular, round. And most of those are going to be water soluble or not water soluble? Water soluble. Water soluble. They're floating around, they want to minimize their contact with the solvent, so you make more of a sphere shape. They're not perfectly spherical, but they're certainly not like that, they're long filaments. That's the other type you can have is filamentous, like this, right? So they're really long or fibrous proteins. Those are more likely to be water soluble or not water soluble. Not water soluble, right? So they're really, really long. Even if they're covered in hydrophilic things, they're just too big to be in solution, right? They're just too long. They're thousands of amino acids long, so it's just too big physically to stick with solution. Okay? And then, for example, this is keratin, right? So keratin has these coils, right? You see it's two alpha helices. And I said they have that slight twist to them. Right? If you make it long enough, you can observe the twist. Well, these two are twisting around one another over and over. This should remind you of a structure we use in engineering. It makes it stronger. No, just a simple structural feature that you make. Instead of putting a big cylinder of steel there, what makes a stronger cable than just using a cylinder of steel? Many, many wires wound and compressed together. Because if any one of them fails, it's okay. Right? But how do they not fail? Why would a big solid steel cylinder be better? Because if that forms a crack, it goes right through the whole thing. The whole structure fails. So we wind many of these together, and because they're twisted, they can actually be compressed and pulled a little bit without breaking. It's bent a little. That's what these things do. These are held together by peptide bonds. If you break any one of them, the other strand can probably hold it together. We need to go with peptide bond. So if I twist these together, it gives it much more strength. And that's the keratin found in your hair, your teeth, your bones, everywhere else, to hold things together. And the other one down here is collagen, also structurally holding things together. And it's a triple helix. Right? It's not I don't want to say it this way, it's not three alpha helices put together. That's not what I mean. That is two alpha helices put together. This doesn't have an alpha helix in it at all. It's three strands that are forming one helix. Right? It's just called a triple helix because there's three strands twisting around each other. Does that make sense? This is one strand twisted on itself at the top, another strand twisted on itself, and then the two of them entwine. Right? Like two separate cables put together. This is forming one cable, but with three strands. Okay? So if you're going to put three of them together, you see the space in the center there would have to be really small. Because one of the R groups has to face the center if you put three of them together. If you just do two, all the R groups you told me face out radially, which makes sense. It's twisting on itself. But if I put three together like this, one has to face the center, and there's only one amino acid that'll work. It's got to be the smallest one. Right? Only glycine works. Right? No amino acid R group out there, just the hydrogen. That's the only one that'll actually form this thing. So collagen is, all along the way, plenty of glycines every time it faces the center. Does that make sense? And in fact, I actually don't want it to form a helix, because then it won't be able to do this. So the pattern for collagen is we do proline, proline, and then a glycine. Proline, proline, glycine. Proline, proline, glycine. Occasionally we'll put something else in there. Right? But it's proline, proline, glycine. Proline, proline, glycine. Right? So if you do that, is this going to form an alpha helix on its own? If you have just a sequence, PPG, 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 over and over and over. Proline is terrible for alpha helices, you remember that? And glycine is terrible for alpha helices. There's no chance this will make an alpha helix. Because it contains nothing alpha helix worthy. And so this will only form this triple helix. That's what collagen is. Collagen again connects all the different parts of you together. So without keratin and collagen, you would fall apart. Because you have cables holding this together. Right? And then the third group, I said, globular or fibrous, is the other one could have any shape really, but it's not found floating around in cytoplasm. It's not so big that it can't be soluble. It's forming cables. It's found in membranes. So it's kind of an inside-out globular protein. And so if you think of a globular protein, on the surface, what type of amino acids would you want to put? It's soluble in water. Polar, charged, hydrophilic stuff, right? And what would you put towards the interior of a globular, water-soluble protein? Hydrophobic stuff, greasy stuff, right? The isoleucines, leucines, valines, methionines, those things all in the center, right? So when it folds, those end up in the center and the soluble stuff's on the outside. A membrane protein is kind of that inside out. What will be on the surface of those? A lot of hydrophobics, not exclusively. You probably want one end that has some hydrophilic, so it knows which side to cork up in the membrane, right, to face the phosphate in. But many of the hydrophobics, so it's interacting with the lipids in the membrane. So it's kind of an inside-out protein. This is the example of vitamin C. I'll skip the story for vitamin C. I think it's in the recording, right? You guys can watch it from there. All right. Like I said, globular protein is one on the right, carbonic anhydrase. We'll talk about that enzyme later, what it does. But you see, it's kind of a spherical protein. Right? In the very center, there's a little sphere there of white. That's a metal ion. We'll talk about what metal it is later. But there's a metal ion there that the protein has to interact with. And it's not serving any purpose as far as the chemistry goes, at least not at the moment. But it's serving a place where other amino acids can interact with the metal and hold themselves together. Right? So what amino acids would interact with that metal? You have to think about the metal. The metal is likely to have a charge, right? What charge would it likely have? Positive. Could be monovalent, could be divalent, could be hexavalent. We don't know what it is, but it's definitely going to be positively charged. It's unlikely to have a negatively charged metal ion. Right? So what would interact with it? Yeah, negatively charged things, exactly. So your acid amino R groups, right? What else might interact with it besides the aspartic and glutamic acids? Any other R groups that would do it? Those are obviously the most common.
Could be carboxylate.
Yeah, those would be the carboxylates, right? Any other group that can act as an acid?
Oh, it would be tyrosine.
Tyrosine can, right, to a lesser extent, right? And then one that could really interact with it, not so much as an ionic interaction, but a covalent. Give you a hint. Anybody in here ever broken an old-fashioned mercury thermometer? Ever seen one? Right? They use like red dye colored ethanol in them now because it's much safer. But the older ones have mercury in them, right? If you ever spilled one of those in a chemistry lab or a physician's office or something, how do you clean it up? No, I'm glad you've never had to clean one up. But if you spill mercury, you clean it up with powdered yellow sulfur. Right? You get the bucket of sulfur, right, or the vial of sulfur in the physician's office. But in a chemistry lab, you get a bucket of yellow powdered sulfur. You take it and you pour it on it, and the sulfur binds very tightly to heavy metals, like mercury. Right? So what would bind very tightly to this metal, no matter what it is? Assuming it's not sodium or potassium. Cysteines, right? And so it would actually lose its H and form a covalent bond to the metal from the sulfur. It binds really tightly to metals. Okay. So you may have some cysteines involved. Histidine could also do it because it can have an interaction with the nitrogen to the metal. Less likely than the others, but it can happen. Depends on the metal. But anyway, this thing's kind of round, right? Globular. So the center isn't so hydrophobic as you thought. We can substitute other things to make it hold that shape. So don't think only one way matters. By far, most of them have a hydrophobic core, yes. But this is one example where it doesn't. And then those in the membranes down here, we got the different groups. So I like to think of these as band members, right? So like 1960s traveling bands, and you had the groupies that hung out with the band, right? Maybe we frown upon their activity or something. But anyway, some people are in the membrane. Those are the band members, right? They're actually in the band. They're in the membrane. So we call those integral membrane proteins. They're integrated into the membrane, they're part of the band, right? So that's like this guy, this one, and this one. They're actually in the membrane. They don't have to go all the way across, Maybe they're more important to the band and they go all the way across. Those are transmembrane proteins. But they're also still integral. So those three, this one, this one, are all integral membrane proteins. Right? They're in the band. And then you have some that just hang out with the band members. Right? These guys. Right? They're membrane proteins, not because they're in the membrane, but they hang out with those who are. Right? They're kind of the groupies that hang out with the band. Right? Or they're agents or something. Right? So those are one right there, and this right here would be called peripheral membrane proteins. They're not in the membrane. Right? They can't associate with the membrane directly, but they can with those who are. Does that make sense? Right? So integral, they're integrated into it. They could be transmembrane, right? Go all the way across, or they could just be a regular integral membrane protein. And then you have those peripheral ones who are associated with the integral proteins. Okay. Right. And sometimes, if it's in a membrane, it goes all the way across like that, it can have a channel to it. Right? I don't mean an alpha helix going through that has a channel to the center of it. That's way too small for that. This is a many, many pieces put together. It could be, let's say, 7 different alpha helices coming together. And throughout the structure, there's a channel for stuff to get through. That's all that is. Right? It's not just one alpha helix. So there can be many ways to do it. You saw that beta barrel we saw earlier. Right? It would be a big bore through it, right? Big hole through it. Or you can have smaller channels. But they can have channels. Right? The channel would be lined with what amino acids? What would line the surface of the interior of that channel? Like right along here. What type of amino acids might you find along here? Right, so this one, what would you find on the ones interacting out here? Right, this thing has a surface, right? It's interacting with these lipids out here. So out here you find what? Hydrophobic ones. Maybe right around here interacting with the surface phosphates, they'd be hydrophilic. Same thing down here. That kind of keeps it where it is in the membrane. It's not going to tumble or roll over. It's going to stay right where it is. Now it might move laterally, right? But there's no way it's going to move out of the membrane because it's interacting with all these different things. But lining this interior channel would be what? Probably hydrophilic stuff, right? Because if it's hydrophobic, these things could just go through over here. So these are likely hydrophilic things like metal ions or small molecules that are soluble that can go right through the channel. Some channels may have gatekeepers, right? May not allow everything to go through. If they allow anything and everything to go through, those are called porins, and they're dangerous. Right? Some organisms put them in other organisms and go, all right, everything leak out, you're dead, right? Because I wanted all the raw materials. That's what they do to each other, right? Or it could be very selective and only let calcium through, or sodium through, or something like that. All right, any questions on tertiary structure? Right, we looked at the overall shape. It was either a membrane protein, it was a soluble globular protein, or it was one of those fibrous or filamentous proteins. Yeah.
With the barrel, would it be multiple barrels going around it or just one barrel?
For this thing, likely the beta barrel one is just one, because they're long enough to span the membrane. They're huge. Yeah, that would be many, many beta sheets. Usually it's 12 or 13 sheets coming together, sometimes 11, but it forms that twisting on itself. Right. And some of those are water soluble. Don't think beta barrel means only in a membrane. It can be completely water soluble. And it's like a molecular tin can. Right? So it can fold on itself, and it looks like it was like a cylinder with the ends open, but all those loops on the end could close the caps and make it a tin can. It's not made of tin, right? So it's made of protein, but it could be an enclosed space. Those of you in lab, that's the protein we're working on, right? It forms that shape, this one right here. It's actually that protein right there, right? So that is green fluorescent protein. It forms this tin can. That's the one that you're making a mutation in. Okay. All right, now here we have, so we went over primary, secondary, and tertiary structure. Between secondary and tertiary, there's something called supersecondary. Right? What that means is it's not secondary, because that means alpha helix, beta sheet, loop. Those are my only options. Tertiary is the entire protein. What does it look like? Well, we're going to look at a region of a protein. And it's not just one alpha helix, it's not just one strand or one sheet. It's got some things going on, like this. It's an alpha helix, a little turn, which is a loop, and then another alpha helix. And that pattern or that motif occurs often. Right? We call it a helix turn helix motif. Right? Motifs are just patterns you see over and over and over. Right? And if we see them enough, we give them names. So we have a helix turn helix or helix loop helix. Turn is got a turn and loop because it actually is a turn. It's not a beta turn. And then we have coiled coils, you saw that earlier, with the two of them floating around each other, we see that all the time. And we also have helix bundles, this is a 3 helix bundle, but you could also have a 4 helix bundle, and also 6 helix bundles, right? Because they cusp into each other with their curvatures. Right? So these are common motifs, what they're called. It just makes a pattern you see over and over. Even if you see a big protein like this, inside of it, I see this pattern. There's a helix, turn, helix pattern. Right? That motif is there. Those are super secondary. They incorporate multiple secondary elements, but they themselves are not the whole protein, so they can't be called tertiary. Does that make sense? They're more than the sum of their parts, but their parts are secondary elements, but they are not yet the entire protein. Right? Here's another helix, turn, helix over here. Right, we see that pattern all the time. The other one is called domains. Now this one really isn't in the hierarchy, right? So we have primary, secondary, super secondary, tertiary. Domain is a separate category. It's not in the hierarchy at all. What a domain means is, if I look at the whole protein, like this one down here, this protein is one strand from start to finish, amino side to carboxy side. And it has some secondary elements like these beta sheets, right? Fill out the helices in this one. There are over there. But this one's just some secondary elements. And the whole thing, if I look out and call it by its three-dimensional or tertiary structure, it's kind of like groups of globules, right? It's like beads on a string. Think of it that way, right? Like big pearls on a string or something, right? So this is probably soluble in there. It's not a big filament, it's not in the membrane. But it looks like each part kind of folds up independently. You see that? Right? So each one of these parts, they're not separate proteins, it's one strand. But this folds up independent of all those other parts. Right? So we call this a domain. It doesn't need those to make it structure. Now, if a strand from over here came back over here and did something and then went back, that wouldn't be a domain then. It needs other parts from other pieces. Does that make sense? So if it folds up independently, compact unit, kind of separate from the rest of the strand, we call it a domain. It's not tertiary, it's not secondary, it's none of those. It's not in the hierarchy. It's just called a domain. Okay with that? Now, if this had been one strand, and then that's the end of the protein, this would be another protein here, and that's a third and a fourth protein, and they all come together, those aren't domains, those are individual proteins. This would be its entire tertiary structure. Okay with the difference? So what do we call those things? If we have all these pieces coming together, multiple proteins coming together to make the final product. Quaternary. A quaternary structure, right? Do all proteins have that? No, because some of them, like this one, it's just one strand. It has to be multiple strands to have quaternary structure. Right, so these are all examples of folds and domains. Right, let's do quaternary structure. There it is. So we have multiple chains coming together. I'll look up there on the right. This is hemoglobin. Right, there are 4 separate chains that come together, 2 of one type and 2 of another, and they form the hemoglobin molecule, or hemoglobin protein. It does have tertiary structure too. Each of those individual subunits has tertiary structure. They're quite globular, but come together to form the functional unit. Myoglobin, on the other hand, only has one chain. It looks like any one of those, but it doesn't interact with any others. Right? So it would not have quaternary structure. We call it a monomer. It's only got one part. Mono means one, mer means parts. Right? So monomer is part. This one has how many parts? The one at the top. Hemoglobin. It's got 4 parts. You call it what? Tetramer. Exactly. There's more than one way to make a tetramer. We'll talk about that in a minute, but that's definitely a tetramer. All right, so here's some other examples of quaternary. How about the one on the top left? Those are 2 identical proteins, one in red and one in yellow. Right? You notice they're not the same strand. Right? They're 2 separate strands that have come together and associated in this way, right, to make the functional protein. But they're identical to each other. It's just 2 copies of the same protein. Does this completed unit have quaternary structure? Yes, there's more than one copy there. In fact, there's 2. So it'd be called a what? A dimer first, right? Because there's 2 of them. But what is the 2? I need to tell you, are they identical? Or are the 2 different proteins that came together? So I need to preface it with some kind of prefix, right? So we say they're identical, we call it a homodimer, meaning same. Or a heterodimer, meaning different. Makes sense, right? Easy to remember. So So this one has two identical subunits, homodimer. This one over here is another picture of our hemoglobin. We said there was two of one type and two of another type. So it's called a tetramer still, but it's a heterotetramer. They're not all the same. The only way to get the homo prefix is to have all copies the same, however many there are. If there's 8 copies and they're all identical, what would you call them? Homooctamer, right? Because they're all the same. If there were 8 things there, 7 of them were identical to each other and 1 was different, it's a heterooctamer, right? Now, you probably figured out there's more than one way to do that. So I can't just call it that, I need to be more specific. In this case, I label what they are. It's like a little, like chemical notation, like, H2O or something, right? You would say how many of each type there are. 2 of the alpha type, 2 of the beta. And we generally just give them Greek prefixes, right? Doesn't matter which one you call alpha, which one you call beta. The literature kind of sets itself on one and everybody follows it after a while. But for a new protein or for one you're just evaluating, you can call any subunit alpha, beta, gamma, delta, and so forth. Just give me the list. It's like a chemical form. Okay? All right. Here's some examples. So let me remind you real quick before we do this question. What held primary structure together? The peptide bond, which was chemically amide bond, right? That's it. Right. And what holds secondary structure together? Exclusively hydrogen bonds. Between what and what? Well, that's the combination mathematically for alpha helices, but what 2 things are always involved? The hydrogen bonding between what and what? The carbonyl on the backbone and the nitrogen on the backbone, right? That's it. No R groups involved. What about tertiary structure? What holds it together? Everything. Anything you think of, right? So, hydrophobic interactions, which are weakest, right? Hydrogen bonding between R groups and other R groups and backbone, whatever you like to think of for tertiary, right? Anything. Disulfide bonds, ionic interactions. Anything that can interact is plausible for tertiary. Okay, so easy way to remember that. Anything possible. Not the peptide bond. That's what held primary together, right? That's not holding the tertiary structure together. And then what holds quaternary structure together? Anything that could have held tertiary together except disulfide bonds. It's disulfide, that's not quaternary structure. That's interacting covalently. So non-covalent interactions hold quaternary together. Alright, so speaking of that, let's label these with their letters. So A says beta pleated sheet. Which one of those does that represent?
Secondary.
That's secondary structure, right? The order of amino acids in a protein, what would tell me that? Primary. A protein with 2 or more peptide chains. Primary structure, that's the definition. Shape of a globular protein. Tertiary tells me that. And a coiled coil of alpha helices. That's not secondary because it's a combination of secondary elements, so it's super secondary, but it's not yet the whole protein. So it's not tertiary. Make sense? Okay, 2 things I can do to my folded protein to destroy it. I can add some agents or treat it in certain ways that all these structures unfold, right? And everything falls apart except primary structure. What would that look like if I did that to a protein? I break every level of structure except the backbone, except the peptide bonds. It just kind of unfolds via a long string, right? What do you call that? Right, the primary's left. What do you call that process? Denaturation. It lost its nature, it lost its fold. We already did this to DNA and RNA, remember that? It's the same thing, they lost their structures. We didn't call them secondary structures, but that's what that was when it was in the helix. And it just became a string. Right? Now that was easily reversed in nucleic acid, they just came back together. This is easily reversed in protein. So the interactions are far more numerous, right? So you can't unfold a protein and expect it to fold back easily. In fact, almost none of them refold easily. There's a few, you probably can think of 2 qualities about the proteins that you can refold. So if you're going to refold one after denaturing it, it's really hard to do. What properties would you want your protein to have to make it easier to refold? Small is good, right? If it's huge, this is going to be unlikely, right? It's too many combinations. So a small protein, that's good. Like what else? That's the primary. What else? Would you want it to be a membrane protein or a globular soluble protein?
globular soluble kind. If it unfolds, it's probably gonna precipitate out a solution, right? Because you're exposing the hydrophobic core. But if it's a membrane protein, you do that, it's gonna now be soluble, and never get it back out of solution. Denatured it to get to that. So it's easy to unfold them, but if you slowly get rid of the thing that's denaturing it, if it's a detergent, or if it's a temperature, or if it's salt, or whatever it may be, you allow it to refold slowly, you might get it to refold. But it has to be small, so there's not many other combinations of things. And it has to be easy to do. Right? So it has to be, you know, favorable, only make these three, you know, ionic interactions in the cores together. Something simple. Very good. All right. So if I do that denaturation process, I break every level of the hierarchy except one. Primary still. How can I break primary? Add an enzyme, and it does what to it?
Hydrolysis.
Hydrolysis, yes. So I do protein hydrolysis. So I'm gonna break the backbone. So if I do protein hydrolysis, what levels of structure does that disrupt? Primary. If you disrupt primary, you disrupt everything above. Just like if you disrupt tertiary, you automatically disrupt your quaternary. Right? Any level of structure you disrupt, you automatically lose all the ones above. Makes sense. So denaturation destroys or disrupts everything except primary, and hydrolysis destroys everything in structure. Let's talk about this one real quick, and then we'll take a break. All right, so these are called prions, right? These are proteins that can be infectious. And you think, wait, it's not a living thing. Right? It's not. But they can propagate themselves. There are many non-living things that can propagate themselves. Think of another example. Fire is the most common example, right? Is that what you said? Yeah, fire. It propagates, right? It's not living. It doesn't meet all the qualifications that we put for living things. It has no genetic memory. It has none of those things. But this protein can do this too. Right? So if you take a protein that looks like this normally, it's rolled up, have these two alpha helices here. Right? See, this is a forward helix bundle. These two back there kind of interact with each other. These two are folding like this. The two in the back are covering like that. So nice stable protein. But if you disrupt this a little bit, right? You don't totally destroy it, you don't totally denature it, you don't totally hydrolyze it, but you disrupt its structure a little bit and allow it to refold, it might refold improperly like this. So I converted these two alpha helices into a base difference here. Makes sense? And it's still stable protein. But because I disrupted it, if you looked at this protein, what would you imagine is on sticking out right here, right here, right here, right here, but not in the ones pointing in. These are all arteries, right? Pointing outward radially as we go around. The ones facing the outside might be what? Like your filling stuff, polar charge things. And the ones facing the interior of the other helix, the ones from this helix facing this way, probably hydrophobic to hold it together. Now I've completely unfolded these two helices and refolded them like this. Do you think everything facing out is still hydrophobic? Or sorry, hydrophilic? And everything facing, remember it's facing every other one opposite way. Right? Do you think they're still in that arrangement? Highly unlikely. Right? So the ones facing out might be more polar or less polar than they used to be. So this protein, what it does is it misfolds like this. It finds other proteins like this, and gets next to it, binds to it, which forces these to refold like that, to copy themselves basically, and it makes these long fibers like this. And so normal soluble folded stuff, right, don't interact with each other because they're all isolated properly. If one misfolds, it causes the others to misfold and form these long fibers. Right, because then they can pack their hydrophobic surfaces and not interact with each other. Right, and those aren't good because they'll form plaques like this. Right? This happens in your finger, your toenail, no one cares. Right? The body's like, whatever, we'll destroy the protein and make a new cell. What if it happens in a neuron? We don't throw those cells away, right? We don't replace them. So if this happens in a neuron, those cells die. And you don't replace them, and you get these plaques in the brain like this, and it's non-functional. Right? This is the cause, or one of the main causative agents of a lot of our illnesses like Parkinson's, Alzheimer's, and things like that. It's not the only cause, but it's one. Right? These are called prions. So how does this happen? How do we get a partially unfolded, refolded protein like this?
DNA.
If you damage the DNA, that would change maybe This has the exact same sequence. It's just been folded properly at some point, disrupted a little bit, not completely digested like you would if you went through a digestive tract, but sort of unfolded, refolded incorrectly, made its way to the brain and caused this problem. It's not likely to happen, but it can. Well, it doesn't reproduce and make more of itself. It makes more that look like this into ones that look like itself. These are already there. It just convinces all of its neighbors to look like it because it makes it more stable in the fight. So once one gets started, it's a chain reaction. Usually stops when it kills itself. Some of those debris make their way to neighboring cells and then start to. Yeah.
The chaperone proteins, like, do they have anything to do with like prions, or I'm just curious about that?
Well, this protein could be pretty much any protein. Right? It doesn't have to be any particular type. It just has to be one that will have this feature. It's not always an alpha helix to a beta sheet type, it's just one example. But it's one that misfolds somehow after partial denaturation. And when it refolds its improper way, it causes the others to do the same. Yeah. What do you think?
I was just saying, like, because like some proteins, these chaperone proteins help them fold sensitive. Like you asked, like, well,
Right,
why
so
does that
maybe-
have to turn to that? So I was just asking, well, maybe, I don't know.
Then you probably wouldn't get a folded protein at all, right? But this is more subtle, it's more insidious. This folds properly, right? And then you get one somewhere that isn't folded and causes others like it to misfold. Yeah.
So the original one that was folded, if it makes the other ones, I guess,
Misfolded as well.
misfold as well.
Right.
If the original one, dies or degrades, will the other ones go back to its original, or will they
Now,
continue?
if you make all this and then take, say, one off of here, this is still gonna keep propagating, right? So it's kind of a one-way street. But this had to get introduced somehow, this misfolded protein. Any other guesses where it came from?
So is it just something as simple as like as you age and throughout the years you just have a higher chance of
That's
something
one
going
thought
wrong?
behind it, right? That as proteins degrade, somehow it gets misfolded. And that's one aspect of maybe some of these diseases. But there's a faster way to cause this, and we know it's the cause. The aging might be one of the reasons this happens, yes. Right? But there's a more direct cause that we know is the reason.
If you, so, these things, is it something like, honestly, like,
He's smiling, he knows what it is.
is it like disrupting
I'm
like
glad they don't know this.
the signal or something, like you're disrupting like the,
Yeah, silence of the lambs.
so,
Cannibalism.
Oh, yeah, back disease.
So how would you get a partially misfolded protein that looks identical to yours? Cannibalism. And so, now which proteins are we talking about? Where are they from? Right, so if you're eating other human brains, right, which is not a good idea, right, and you didn't cook them all the way, also not a good idea, right, you could partially get this misfolded protein in your diet, right? Let's say for some reason it doesn't get fully digested, it makes its way through the bloodstream to your brain, it could cause cancer. Right? Now that seems absurd for humans, right? We force this on some animals, right? We feed ground up beef to cattle. They're cannibals now. We made them into cannibals. And this is what causes things like cow disease, right? Or in humans it causes what's called Yago disease. And because the bovine protein, the cow protein, is almost identical to our human protein, if we eat that, we can catch the same thing. Right? It was first discovered in sheep. It was called scraping. Right? Where the sheep they're feeding sheep other sheep, right? That's what they were doing. Bad practice. Right? But they didn't know. Right? But they were feeding it to them, and some of these sheep were going nuts because they're losing brain cells, right? And they would just start scraping themselves along anything, everything, wires, buildings, roads. So they call it scraping. Like, kill those sheep, get rid of them. Don't grind them up and feed them to them. They didn't know that's what happened. It kept spreading, spreading, spreading. But now we know it. And yes, maybe aging is responsible for some of these misfoldings. Because technically, we're not supposed to live beyond maybe 30. Our job as reproducing is done, right? From an evolutionary standpoint. So if we live to be 90 to 100, it's very likely we're causing some misfolding. If it's not in brain cells, we're okay with that. We'll replace that with a cell. But these we can't replace. tried to prevent that. It's hard to do, to police every cattle farm across the planet, right? Some of them are less, let's say, less concerned with it than others. And occasionally you'll see this pop up, like usually in England, right? It pops up a lot of times because someone did it and they slaughter all the cattle.
And actually, partially because of that England incident, they've been a lot more careful since then. There's a lot more regulation floating around with that type of such a lot.
Yeah, there's so many regulations in place, but in some parts of the world, let's say in South America, where there's a huge cattle and beef industry, the oversight is not there as much, so this may go unreported. We don't report those here as well. What's that?
I was more concerned about the US.
US is very strict. So if you're gonna be a cattle farmer here, it costs so much more money than it used to. Yeah.
So you said the prions came because people ate people, I guess, right? Did they eat the people right after they died, or did they wait?
Well, it's
for a
not
while?
necessarily human cannibalism. It's the reason we see like Creutzfeldt-Jakob disease in humans is because they're getting this exact same protein. The bovine one is identical to ours in sequence. There's no difference. So we call it a bovine protein, but you couldn't tell it apart from human.
I'll probably ask you later, I'll do it.
And that's the same with other organisms like sheep and pigs and what else?
Chickens can get prions.
Chicken's completely different. Alright, so if you eat chicken, you're good. So this is why organisms should not eat other organisms that are closely related